Okay, so this is my second video on pancreas, and today I'll be talking about insulin. Now, within the pancreas, there are about 1 to 2 millions of islets of Langerhans. Now, within islands of Langerhans, there are three types of cells: alpha cells, beta cells, and delta cells. Now, the alpha cells produces glucagon, beta cells produces insulin, and delta cell produces somatostatin. Today, our focus is mainly on the insulin. In the previous class, we had discussed about glucagon production. So let's do a quick recap. So pancreas, they are the heterocline glands. So we have an exocrine part and an endocrine part. Now within this endocrine parts, it has islets of Langerhans, which consists only 1%. Okay. Now here we have alpha cells, beta cells, and gamma cells. Now the alpha cell produces glucagon, beta cells produces insulin, and delta cell produces somatostatin. Now insulin is produced when there is hyperglycemia. Now, what is hyperglycemia? Hyperglycemia means when there is huge amount of glucose present in the blood, which is more than the normal range. The normal range is 80 to 120 mg per dl. Now, if it increases more than 130, actually if it just increases about 120, it's still quite normal. Okay, so when it reaches above 130, then we call it as hyperglycemia, and in that case what will happen, insulin will be secreted from the beta cells. Okay, in the previous video I was talking about this glucagon synthesis and the function. Similarly here I'll be talking about the synthesis and production and also the function of insulin. Okay, insulin is produced when there is hyperglycemia. That means when the blood glucose level is more than 130 mg per dl of blood. Okay, so out here my glucose level is very high. So that means there's a feeding state. When we are eating, the glucose in the blood is high. So in that stage, what will happen? The glucose from the blood will enter inside what? Beta cell. I've drawn her a beta cell. Okay, beta cell of islet of Langerhans. So what will happen? Glucose will enter. Now the receptor that is used for this is called as the GLUT2 receptor. Now what kind of receptor this is? This is an insulin independent receptor. Now what does that mean? It means this is such a receptor which does not require the presence of insulin to be open. Okay, so in order for it to function, it does not require insulin to be there. So that means even if the insulin is not there, this particular receptor will be open and will be functioning. So it will take up glucose even if insulin is not present, right? So now here, since in our blood the glucose is very high, so very high amount of glucose will enter inside the cell, inside the beta cell. Then what will happen? This glucose will be converted into glucose 6-phosphate, then into pyruvate, then into acetyl-CoA. Then what will happen? This acetyl-CoA will now enter inside mitochondria. So I'll just draw with this. Okay. So this entire thing will happen inside a mitochondria. Okay. So it will it will become acetyl-CoA, then will enter a Krebs cycle, and everything will happen inside. Suppose this is a mitochondria. So it's happening inside a mitochondria. Okay. Now inside the mitochondria, all these things will happen. And then out of this, what will be produced? FADH2 and NADH. Now FADH2 and NADH2 will then be converted into ATP. Now what happens? This ATP will bind to this particular receptor. Now when this ATP binds to this receptor will be switched off. Okay. Because of that, what will happen? The membrane potential will increase and therefore it will become positively charged. So basically, the amount of potassium is increasing because it is not able to go out. Okay, so because of that, what will happen? It will affect this particular channel. Now, what is this channel? This is a calcium channel. Now, what happens because of this? All the calcium which was present outside the cell will now start entering inside the cell. Okay, so calcium will now enter inside the cell. Okay. Now, let's see what else is happening inside the beta cell. Now, beta cell has got its own DNA. So, basically, this is the DNA inside the nucleus. Now, there are specific gene out here which is responsible for the production of insulin. So, what will happen? This DNA, the gene that is supposed to produce insulin, will get activated and therefore it will produce an mRNA. Okay? Okay. Now, this mRNA There will be some ribosome, and then it will go for trans. After translation, it will make a protein. Now this protein is called as pre-pro hormone. Okay, this is produced. Now this protein will enter inside the rough endoplasmic reticulum. Okay, where inside the rough endoplasmic reticulum it will go through some kind of modification and all. And after that, now this protein will be called as a prohormone. This is still not a hormone, all right. Then this will be packaged and it will send inside the Golgi, where vesicles will be formed and further modification will take place. Now from the Golgi, what will happen? It will be packaged. Okay. So within Golgi, it will be packed. Okay, packed within the vesicles. Then what will happen? These vesicles will come out of here. Okay. Now this vesicles, it contains insulin. Okay, so here I have insulin. After the final production, we now got the hormone. Okay, now along with this insulin, it also contains another thing out here with insulin. C-peptide is also present. Okay, insulin and C-peptide, both the things are present. Now what happens? Because of the presence of this calcium, these cells will fuse, not cells, these vesicles will fuse, and therefore the insulin and the C-peptide will be released. Okay, and where will they be released? They will be released in the blood. Okay, so now the insulin and the C-peptide is now present in the blood. So insulin, C-peptide, okay, C-peptide and another thing, amylin. All these things are coming from the beta cells. So let's recap once again. So what's happening in the beta cells? So beta cell is taking up blood glucose, okay, because it's very high because we are feeding, and the blood glucose will enter and will convert it into glucose 6-phosphate, then pyruvate, then acetyl-CoA, and it will enter the Krebs cycle. From the Krebs cycle, it will produce FADH2 and NADH, okay, energy molecules. It will be converted into ATP within the mitochondria. Then because of this ATP production, it will bind to this particular receptor, and therefore the membrane potential will increase. Now, since the membrane potential is increasing, it is the cell is becoming more positive, and therefore the calcium channel will open, and because of which the calcium will enter. Now, what is function of this calcium? This calcium will help the vesicles to fuse. Now, what are these vesicles? Now, from the DNA, there is a gene which is supposed to produce insulin. It will produce mRNA. From the mRNA, protein will be produced. It will go through several kind of modification within the rough endoplasmic reticulum and the Golgi, and then the vesicles will be produced containing the insulin, C-peptide, and the myelin. Okay. Then this calcium will help these vesicles to fuse with the cell membrane, and therefore insulin, C-peptide, and amylin will be released into the blood. So this was the synthesis of insulin. Now we will see the function of insulin. Okay, what it does. So the first, what will happen is, so in my blood, the glucose level is already very high. Okay, and also I am having insulin present out here. So I'll draw insulin like this. Okay. So in my blood, insulin is there, and this the amount of glucose is also very high. Now, this glucose will enter inside the liver cell. Now, again, there is a GLUT2 receptor, and GLUT2 receptor is again insulin independent. So basically, it does not require the presence of insulin to be active. Okay. Now, so the glucose is entering inside the cell. Now, what will happen? When the glucose, after the glucose is entered inside the cell, the glucose will be converted into glycogen. But this process is helped by a certain thing. By what? We'll see. Now, what happens? Out here, we have this insulin. So, the insulin will also come out here and it will bind to a certain receptor. Now, this receptor is TKR, that is tyrosine kinase receptor. Okay? With this tyrosine kinase receptor, it will attach, and after attaching, it will give this second messenger, protein kinase B. Okay, protein kinase B, and there is another one, PI3K. Okay, after it has produced, this will go and it will give a positive signal so that the glucose can be converted into glycogen. All right. Now, this particular process of converting glucose into glycogen is okay. So this is called as glycogenesis. Okay, glycogenesis is taking place where glycogen is produced from glucose. Okay, what else will happen? The glucose will also be converted into pyruvate, pyruvate to acetyl-CoA, and then from acetyl-CoA it will go to the into the Krebs cycle. From Krebs cycle it will enter the electron transport chain. From the electron transport chain, finally we will get some ATP. So because of the glucose, a lot of ATP is also produced within the liver cell. Okay. Now the process where the glucose is getting converted into pyruvate and finally the ATP, this particular process is called as glyco glycolysis. That means a breakdown of glucose. Okay. So these two processes are taking place within the liver cell. All right. Okay. So these are the main processes that takes place within the liver cell. Now, let's see what happens within the muscle cell. Now, in muscle cell, what happens again? I have high glucose level within the blood. So, glucose will come from the blood and it will enter inside the muscle cell. Now, out here, I have written GLUT4 receptor. Now, within the muscle, the receptor that we have is called as GLUT4. Now, what kind of receptor this one is? This one is insulin dependent. So that means this particular receptor is only going to be open when insulin is present. So that means the presence of insulin is very important for it to be open. So that means, so let's do that. I'll give it some insulin. The insulin is coming from here. Okay, so this is I, insulin. This is I. Okay, so insulin is coming from the blood. Okay, now because of this, what will happen? It will activate protein kinase B, and also we have this PI3K. Okay, so this is activated. Now, what this will do? This will give a positive signal to this GLUT receptor, and therefore the GLUT receptor will open. Okay, and after this opens up, glucose will enter. Now, then glucose will convert it into glycogen. Okay, glucose is converted into glycogen, and this particular process is also activated by this protein kinase B. Okay, other than that, what will happen? Glucose will convert into pyruvate, into acetyl-CoA, then will enter into the Krebs cycle, then it will go to the electron transport chain, and from the electron transport chain, finally, ATP will also be produced within the muscle cell. Okay? So this is another process that will take place. And what else? Now, when we are eating a lot of food, that means we are also eating a lot of protein because our diet also contains a lot of protein, not just carbohydrate, protein as well. So that means when you are in the eating stage, the feeding stage, we also have a huge amount of amino acids present in our body, in the blood. Okay, so where will that go? So these amino acids will taken up by the muscle cell. Okay, amino. So basically, what kind of channel this one is? This one is amino acid channel. Okay, so from the amino acid channel, this amino acid will also enter inside. So these are all amino acids, different kind of amino acids. Okay, so these amino acids will then be converted into a peptide. Okay, this one is a peptide. So basically, peptide formation or protein formation is taking place. Protein synthesis is also going on. So what are the processes that is taking place out here? So here we are having within the muscle cells, we are having protein synthesis is going on. Okay, protein synthesis is going on. What else is going on? Glucose is getting converted into glycogen. And okay, first of all, glucose uptake is there. Okay, glucose uptake is there. Glucose synthesis has been increased. What else is happening? Then glucose is getting converted into glycogen, and also there is a process where glucose is going broken down. So glycolysis. where glucose is broken down for energy, ATP. Then there is glycogenesis, that means the production of glycogen. So all these processes are happening within the muscle cell. Okay? Protein synthesis, and also there is uh, so these things are all happening within the muscle cells. All right. Next, it also has function on adipose cells. So these are the adipose cells. Now, what happens within the adipose cells? Again, you can see here I have drawn this GLUT4 receptor. So there's high amount of glucose in the blood. So the glucose will enter through the GLUT4 receptor. Now, since it's GLUT4 receptor, so that means it is insulin dependent. So that means it requires insulin to be present in order to function. So here I'm drawing insulin, which is coming from the blood. Okay. Now, so the insulin has entered. So again, we have this PI3K is also there. Yeah, okay. All right. So this will give a positive signal. So the channel opens and it takes up glucose. Glucose will be converted into glucose 3-phosphate, then into pyruvate, then into acetyl-CoA. Then what will happen? This acetyl-CoA will now produce this. What is this? These are fatty acid chains. Okay, fatty acid chain. So fatty acid chains are being made from the acetyl-CoA. Now, what is this particular process called? This particular process is called as lipogenesis. So lipogenesis is taking place within the adipocyte. Okay. All right. Next, what is happening? This is glucose is also converted into DHAP, which is converted into glycerol. So suppose this is my glycerol. So glycerol is produced. Okay. After the production of glycerol, what will happen? This fatty acid chains and the glycerol, both of them will come together. Okay. And they will produce this molecule which is called as triglycerols or triglycerides. Okay? Triglycerols or triglycerides, which is also called as TAG. So TAG is produced within the adipocytes. Okay? This is the like these are the things that happens. So basically, in adipocytes or adipose cells, what are the processes that is taking place? Out here, we can see that there is glucose uptake happening, okay? And there is lipogenesis happening. So these are the two main processes that takes place within the adipocytes, okay? And the upar wala likha hai, all these things were happening within the muscle cells. Okay, so I will write here as well. So in the liver, what were happening? Glycogenesis happened, glycolysis happened. Okay, these two things mainly was happening in the liver cell. So these are the major functions of insulin. Now within the blood, okay, there's another thing happens. Now within this adipo out here also in the blood when we're eating a lot of food, so we also have fatty acid in the blood, not just amino acid. Amino acid is taken up by the muscle cells. Similarly, some of the fatty acid is also taken up by the fat cells because again our food, because we're eating in the feeding stage. So some of the extra fatty acid is also taken up by the adipocytes and converted into triglycerides. Okay. So these are the major functions of insulin. So in the next video, I'll be comparing insulin and glucagon in another video. Okay, so ultimately, after all these processes, what will happen? What will be the end result? So after we have taken up all the glucose from the blood, okay, amino acids, fatty acids. So ultimately, the result will be, okay, ultimately, the result will be that the glucose, that is the blood glucose, will go down and it will reach the stage between 80 to 120 microgram per deciliter, which will become normal. So that was the end result. Okay, after the synthesis of insulin, and then it has been taken up by this, like, glucose is taken up by liver, muscle cell, and adipocytes.